Today's sections with Professor Fernando Nilo from the
Andrés Bello.
Universidad Andrés Bello de Chile y que nos vas a hablar, no me acuerdo el...
Several things. Don't worry.
Okay, so let's try to keep it under the schedule, 30 minutes.
Yeah.
So we already have 6 off.
Okay, I have to rant today.
All right. Thank you,
But,
Fernando.
hi everyone. I'm really happy to be here. As usual, come to Brazil all the time. It's very exciting. Thank you, Munir, for your invitation. And also thanks to Sergio Pantano, who is beginning with this initiative and was very interested in the meeting of yesterday. Today I will show several things. First, I would like to introduce to you our center that is a little bit, has some evolution during the time. For the reason, I would like to show you how we are implemented, how we can introduce the bioinformatics and molecular simulation in the context of innovation, in the productive sector. For us, the most important is that the innovation is related directly with the multidisciplinary discussion. For that, we have implemented a center where combined in the same place the experimental installation with bioinformatics or computational biology tools. We have something that is working well, is 1,500 core silicon graphic system, a lot of different programs but all of them related with bioinformatics, genomics and structural bioinformatics, database, etc., molecular dynamics, obviously. And we have a small installation for 3D visualization. Usually I say that all my colleagues that if you want to implement a bioinformatics tool lab, the first that you have to buy is a 3D visualization, because then you can combine to your director that you have to buy a big computer. It's the only way. So also inside of our university, we have implemented a pipeline that is a very interesting initiative related with bio-nanotechnology that combine all our progress in biotechnology and nanotechnology in the same context, from the atomics, synthesis, in vitro and in vivo assays. In that regard, we are using as an iterative cycle that we beginning with the experimental event. Then using this event, we make some theoretical evaluation through bioinformatics or molecular simulation. That means that we take this information from genomics, for example, or X-ray information or biochemical information. Then we generate an hypothesis, and that hypothesis has been experimentally validated. This cycle allows to us make innovation, and the most interesting is to obtain new concept. That is really important for us. Our team for bioinformatics is including these 4 labs, microbial genomics, molecular biophysics, bio-nanotechnology, and drug discovery, that are working together with another experimental lab that include bio-nanotechnology and microbiology, biosystem engineering, molecular biophysics, nanomedicine, chemical synthesis, and also we have good collaboration with the Sequence Genomics Center. Among our most interesting research area is Dr. Jose Perez. Usually he makes some exploration in Antarctic, and he takes some extremophiles in order to find new bacteria and specifically for the synthesis or biosynthesis of quantum dot using bacteria. That bacteria has been recently characterized from the genomic point of view and also from the structural point of view. We obtain a very interesting concept related how to decrease the cytotoxicity of the quantum dot that has a promised application in biomedicine, but unfortunately is quite toxic. In other contexts, we have some very interesting discussion inside of our team. For example, I don't know if you know that the bacteria can live at 105 Celsius degree. Thus, it's quite impressive that the bacteria can live there. So how the bilayer can be stable, that is quite hard. In that context, we have some bacteria that has been obtained from in the north of Chile, in the desert. It's very hot, this water. Geyser, in fact, is that. And from this, we implemented a challenge to evaluate what is the property of this bacteria. That is not our work, but unfortunately, well, we are working in the same system. Unfortunately, there was a cover in the Biophysical Journal in the last month. But basically, it's a bilayer that is compounded with several carbohydrates. So for this reason, probably the carbohydrate is very important, huh, for coarse grain. So this kind of bilayer is quite hard to survive to this kind of temperature. And in this context, we characterize some lipid, lipidomics, also called in this area. Well, the bacteria has his own trick. Basically, the bilayer is not a bilayer. It's just one layer with two heads and long tail of carbohydrate. So in that regard, we make some analysis of a structural analysis of this kind of system. And our idea is to use this kind of lipid for liposome for high temperature. That is required in several industries. For example, for food industry, for vaccine, etc. When you produce the vaccine inside of the food of the animal food, usually that system is don't survive and the liposome is broken. So for that, we are thinking to use this kind of system. Yeah, we have a nice collaboration with several synthetic chemical groups. In other context, also, we have a lot of work in genomic, human genome, microbiome. In fact, in microbiome, we have very interesting observations. We are using also this kind of structural similarity network. That is very interesting approach, together with some database of chemical reaction inside of the enzyme. In fact, using this technology, we improve the affinity of some substrate that is very useful for industry that is related with the biosensing, yeah, again to drug. So we have some collaboration with another group more related with genomics. So I not stop here. So in the context of this meeting, well, you know all this guy. All of you are working in some sense. You are reading some of his paper. And in that context, in the context of multi-scale, we have implemented different kind of tools, from quantum chemistry for evaluation of transfer of phosphate in enolate in phosphonol kinase. And here you can see how this proton is transferred to this phosphate, is beta phosphate, in order to stabilize the transition state. So one of our old work in that area. And also we are using quantum dynamic. We have another application that is an old tutorial that we organize in our group. It's basically M2 channels, the proton transport in influenza virus. So it's another nice example how we can use this kind of multi-scale simulation for virus system. Yeah. So here you can see how this histidine is transferred this proton to the water. During the last more or less 15 years, we are working a lot in molecular dynamic of ion channels. In that context, we have many years working on that. Right now, we have produced some rational mutation in order to improve the conductance of this kind of channels. The channel is this protein. It is called the selectivity filter. It is the water. It is the ion, potassium ion. And then you can see in this process, in non-equilibrium conditions, how the ions is beginning to organize in order to make this organization like a line, and then is activated the process of translocation of ions. So Using this kind of simulation take a lot of time, unfortunately, but we have a lot of experimental support in order first to understand what is the role of different amino acids in the potassium ion, and second, also potentially make some rational mutations. Yesterday we talked about the next frontier is the HIV virus, but the good news, I include this photograph here because this guy is Latin American. So maybe we have to invite to the next meeting. That is Klaus Schulzenz. We have a very good relationship with the group, his group, and also with Chris Chipot and other people from this group that during many years visit Chile in order to help us to understand and implement different simulations. In the context of bio-nanotechnology, our focus is related with the controlled drug delivery system. The main problem that we have right now, specifically with antibiotic and other kind of drug, is a unique drug, unique doses, increase the concentration of the drug in the blood, but then decrease very fast. So in order to have the concentration that the drug is working inside your body, you need to multiple doses of the drug. But other option is to use system for delivery during the time and even in the space. So for that, we have some platform with biopolymers. We have Christian Bilo has a lot of experimental experience. He's working with us, and he's implemented this kind of spaghetti. It looks like a spaghetti, but in fact it's a biopolymer. And here you can see a lot of interesting property that has been replicated in this large molecular simulation using coarse grain. In fact, the pore, you can see the pore, the good organization of the polymer that also has been observed experimentally. We suppose that was a mistake from the simulation, but unfortunately the chemists tell us that is observed experimentally. And also we have using low voltage electron microscopy, we can observe how the polymer, the remnant polymer, is the first days is a lot of remnant polymer, but during the day that's beginning to disappear. So finally it is the system with more or less 200 nanometers. Okay. Under this condition, we implement a drug again to cancer cell. So was quite curious because it's expected that this drug has to deliver very fast, because this kind of pore usually produce that fast delivery. But we didn't observe that. In fact, just 3 days later, we observed that delivery of the drug. So something happened with the kinetics. And in that context, we analyze the interaction of the drug with the biopolymer using ABF, or potential of beam force. We evaluate the energy of this drug with the polymer. It is more or less. We observe that the drug has a really good interaction with in the interface polymer water. So for this reason, we can understand that the delivery is only obtained when the neighborhood of the drug is degraded, and then is delivery. Yeah? Using this technology, we implement in cell lines in 3 and 6 pathogens, and we can observe that this kind of strategy is can help a lot to decrease the cytotoxicity of the drug. It is the drug, the cytotoxicity, that is quite high, and it is the system nanoparticle and drug, so decrease a lot. It depends on the patient. All of them are different, and it is only the nanoparticle alone. That article has been well cited. So this technology, taking this concept, with another interdisciplinary discussion, appear another application that could be interesting for all of you. That for this reason, usually I make some parentheses. It is is a patent. This gas en polvo in Spanish. I don't know, powder gas. That is using the same approach. We implement a system for the control of Botrytis cinerea. And here you can see the grape after 40 days that the Botrytis attack the grapes. But here, using our system, the grapes are survived quite well. Also, using another approach, we obtain very interesting result where we can control the ripening of the fruit. In that case, it is the control of avocado. At the end of 9 days, it's not good for the commercialization. But here is our system again that maintain the system under control the ripening. Also, we have used this system in the field with application in apple with really interesting result. And right now right now we are under the process of the commercialization of this product. So the idea is this, that you're taking some approach that use it in biomedicine, you can maybe apply in agriculture. That is something very important in our countries. So in other contexts also, we try to understand what happens when you modify some lipid. It is another lipid, it's a branched lipid, has been synthesized by a colleague in Mexico. He makes some branched region in the head, but he is expecting to form a liposome. But when we observe in the electron microscopy, we saw just some dots, very homogeneous dots. But in order to understand what happened with this system, we use different conditions to organize to synthesize the liposome. But in all the case, finally, we observe just some dots. So when finally Valeria Marquez, who is here, take this work, she make the parameterization of coarse grain, the system, and for when we ensemble all this system in periodic boundary condition, a big system, we observe that also the liposome, this lipid modified, is organized like a ball. The reason is because the lateral interaction is not so strong. So for this reason, it's not generating a bilayer. It's collapsed between them. That is quite interesting because some hydrophobic material is exposed to the solvent. So it's quite rare. Taking the same approach, we are using another kind of lipid. And here we can use this system for self-assembly. We're beginning with a system that has been synthesized. We are working on that. But here in yellow, you can see how this is the hydrophobic part. And the surface of this system, as you can see, in the surface, you can modify. And then you can modulate the interaction with different systems. In this kind of material, another platform or other system that we are using is the dendrimer. We have many years working with that. We create some database. And the last year, we have this nice paper I suggest to take a look. It's a bioinformatic approach for rational design of dendrimer for germ carrier. It is the dendrimer, it's the DNA. The idea is to create a nanoparticle with properties that are quite close to the protein. And for that, in this study, we evaluate all the residues that interact with DNA in the protein data bank. Taking this information, we modify this dendrimer, and Valeria make different combination of residues. It is, in this case, is one of the prototype that we are using, in order to modulate the interaction between the dendrimer and the DNA. That has to be very well modulated because if the interaction is too strong, the delivery of the DNA inside of the cell is not produced. So exist several parameters, biological parameters, to do that. It is just as a reference. Exist several transfection reagents in the market. For example, here we are inserting inside of the cell plasmid that produce a green fluorescent protein. For this reason, you can see some green spot in the fluorescent. It's a lot of cells here. The cost of this transfection reagent is over $7,000. And it is the most popular lipofectamine. Maybe some of you know about that. Here you can see the efficiency of this system is quite good. So the design was done many years ago. Finally, we have the support of some scientific chemical synthesis group in order to synthesize our idea. And it is the first prototype that you can see with a lot of imagination that is quite close to this one. No? We have tried to convince all during a couple of years until finally we consider another approach and we obtain this one that is much better even than the lipofectamine. Here we have a very interesting design. Also, we obtain a better rate of transfections, even more than lipofectamine, that, as I was done in Chile, and finally has been replicated and validated in Germany, in Fraunhofer Institute. So we are very excited with this result, and how also is a nice history how you can use molecular simulation in order to produce a reagent in a rational way. That has been the PhD thesis of Valeria, who is here, just in case. Usually it's quite silent. But even more, usually when you have this kind of result, many people try to find something wrong, right? Happens in all our country. So what about the cost? So probably our cost will be really high. We never produce this kind of reagent. Our country is only good to export copper and fruit. No? So here is the price of the kit that you can find in several companies. Is from 6,000 until 600 or 700 lipofectamine. In our system made in Chile is less than the 1% of any of this system. Even more is $0.35 for one kit done in my lab. So it's, I think that is a motivation for my team and also maybe all of you that we can do several important things in Latin America and in our countries. So just to end, another nice subject for us is the ion channel, as I told you before. One of the channels that we are working is the TRP1. Well, several of these TRP. In fact, we are beginning to work with TRPA1, TRPM8. All of these channels are related with the sensing of temperature. That means that when you put your hand in something hot, this channel, for some reason that nobody knows, this channel is open, produce the translocation of ion, polarize the membrane of the neurons, and then send the signal to your brain and say, take off your hand from this area. So we have channels for high temperature and also for low temperature. But it's very nice to, well, this kind of channel is has a very interesting structure, and how they can be stimulated. Even can be open with toxins, curiously, can be open with molecular organic molecules, and temperature and pH. But in this case, specifically the TRPV1, is also activated with the capsaicin. That is, here it is. Yeah, the capsaicin open this channel, and when open, send the signal to your brain that you have a lot of high temperature in your mouth. For this reason, you feel so hot. Just for your curiosity, I mean, the capsaicin is quite hydrophobic. So to understand how this is introduced in the pocket side of the protein is quite interesting, and also is related with the pain. Just if you have some accident with the kit, the capsaicin is quite hydrophobic, so if the kit filled with chili in the mouth, you can just something like olive oil or something like that. Okay, and if you want has a nice olive oil with chili, you put the chili inside, and then the capsaicin go out. So on the other hand, the TRPM8 that is related with the cold, that still nobody knows how this channel is activated with the cold, but also is activated with the mint. So this kind of channel is very interesting to study in the context of the flavors, but also how you can send the flavors, the temperature, and also the pain. Specifically, TPV1 is related with the broken, the pain when the bone is broken. In that context, we're working on that, but we have to consider that the protein structure of ion channels is a big challenge. It's a really big challenge for us. Recently, it has been accepted in PNS. A long work has been done by Romina. During the last 5 years, we are making molecular simulation with this channel. Is the VK, is one of the most high conductance channels, using spectroscopy assays. Then we can basically measure the distance between different mark between chromophore and lanthanide group that you can install in different parts of the channel. And finally, we observe what is the move, how the domain are moving in the channel. And finally, obtain a model. So obtain a model of ion channel is hard because the identity percentage is quite low, but the architecture is well maintained between the families. But, and today I pretended to visit to a colleague here in Campinas. We have under important revolution related with the cryo-electron microscopy. That technology has been improved a lot. Believe me, it's amazing the number of structural data with cryo-electron microscopy that appear in PDV. Is the resolution. I was in Los Angeles in Biophysical Society. The present resolution until 1.3 angstrom. So they can, using electron microscopy, they can see the hole in the phenylalanine between them. So the revolution of this system has been done because the camera has been improved a lot. It's using astronomic camera. Also the algorithms to take off the noise has been improved a lot. That I think that is the two most important revolution of this system. So during the cryo-electron microscopy evaluation, you have the protein under different position in the sample. It's a very small layer of the solution of the protein that has been very good purified. And then you can select one of these images, and using all this imaging, you can try to find a pattern, 3D pattern, using this system. So as I told you before, I think that we have to be prepared in order to have very good structural information using this technology. I understand that here in Campina, you have a very good installation for cryo-electron microscopy. Rodrigo Portugal, I understand that is one of the experts in Latin America. And so under this advance of this technology, we had the first, was one of the first cryo-electron microscopy high resolution. It is the TRPV1. Before, nobody knows what is the structure, but here finally we have. The resolution is around 3 angstrom. So we have a lot of work under this condition because you have to insert the structure, the atomic structure inside of the envelope of the density, the map of the electron density. And using this information, we analyze how the channel has been opened. Here it is, one of the main change of the structure that we observe. It is one segment, alpha helix, that during the process of open and on the activation of the channel, we can observe how it's broken. In fact, here you can see a long molecular dynamics, and it is the angle that appear in this segment and appear during the trajectory. This system is beginning with the closest system. So for that, we use two molecules that is related with the activation of the channel. One of them is capsaicin, and another one is PEP2. That is lipid phosphorylated. Nobody, during this period, nobody know where is the PEP2. And for that, we make a lot of docking simulation and molecular dynamics, and finally we observe what are the amino acids around to the potential pocket binding of PEP2. And taking this information has been validated experimentally. It is basically the current, and it is the concentration of PEP2. And here you can see how all this mutation produce some impact in the binding of PEP2. Other mutation doesn't produce nothing because are far away from the binding pocket. This simulation is very nice. It is basically, that is capsaicin. The channel is closed. This region is a hydrophobic seal. It is the water ion crossing the membrane. But here, the channel, in this moment, is closed. During this process, you can see the PP2 and capsaicin. And here you can see how the system is open. Yeah? So under this, using this information, then we can understand what is the mechanism, where is transferred the structural energy to this region. The moment we are using another technique, and several mutations are under evaluation. Also using this information and how it's related with the pain, the US Army help us and support this project. Our idea is to integrate electrophysiology with supercomputing. Using our molecular simulation, we know quite well where is the active site of the capsaicin. We understand very well how, what is the main interaction with the capsaicin. And then using a classical docking, massive docking using some database like Zinc, Fluorochem, another one, we characterize the pharmacophore and different potential activator of the channels. Also, we observe that it's very important to understand how this kind of molecule interact with the membrane, because it's a big competitor. I mean, it's very interesting to see that the conformation that adopt the molecule in the membrane is almost the same that when it's interacting with the membrane. So probably the entropy is another issue. So we from the, I don't remember, almost a million of molecules that has been evaluated, we buy 5, and 2 of them, this one, has been is the first time that has been used in that. So it is the correlation between the delta G experimental versus the theoretical, so the correlation is quite good. And also using animal model, we observe that this molecule can affect. Our current challenge is to reconstitute this protein in liposome, as you can see here, and then measure using ITC. That is a nice tool in order to measure the free energy, entropy, and the entropy. So you can validate quite well your system. I know that is Romina, that is a thesis. Just to end, right now we are working in an idea that we try to integrate everything that we call TD5. That is basically the integration of target discovery, basically classical bioinformatics, drug discovery and drug delivery all together in the same context. Well, I would like to thank to all of you, and if somebody has some more specific question, I would be very happy to answer any question that you have. And also, all of you are very welcome to our team, that you have a very interesting team that more than 60 people that is working all together. Thank you very much.
presentation. I will have questions for
each one.
for
Yeah, it's very short time.
on a few ones about the systems that you use for transfection. I mean, the key for being cheap is that it's synthetic instead of that's surprising to me, because I would say that making proteins using proteins to express peptides rich in arginine or lysine will be cheaper than synthesizing the peptides.
Well, we are using a combination of different amino acids that have been modified chemically in order to have a modulated interaction with the DNA. It's working, as you can see, that it's working quite well. Even more, the price that I told you, the $0.35, is 2 times the price that we can buy all the reactive in Chile. So I don't know if it's really cheaper. I think that the pharmaceutical company win a lot of money with this technology.
Do you have limitations for the size of the plasmid that you can?
The plasmid is working very well. We have another prototype for siRNA. That is also working well. And right now we are working with targeting, that the system is conjugated with a protein or antibody or something like that in order to release the drug in the specific site of the organism. Or for example, just for cancer cell, not another cancer, not another kind of cells.
Yes, Maurice?
No.
That was very nice. You showed a lot of success stories of using coarse-grained simulation. And can you tell us a bit about what are the limitations that you encountered in using that?
Well, Sergio is here, so I can't say nothing about the limitation. But from my point of view, the best this system for the self-assembly is working quite well, because many of the self-assembly process is related with the hydrophobic collapse. So in that context, you can see this kind of this kind of event, even with really good experimental validation. I showed to you a small molecule that makes something like a missile. The site is exactly like experimentally. Even more, we make the simulation first, and then another group in the Congress tell us that we are right, it's the same size. So in that context, it's working quite well. It's true that you can lose a lot of specific interaction. You can see hydrogen bond or some specific interaction. So in that context, it's possible to do an inverse simulation. I mean, you can use the coarse grain for the big sampling, yeah, very fast, a lot of microseconds. And then you can, the most interesting configuration, you come back to full atom using the molecule as a cryo-EM surface. You understand me? And then you can integrate inside the molecule, and then you come back to atomic level. That is my experience. I suppose that the hydrophobic collapse is well characterized and well implemented in coarse grain, because Martini, for example, is beginning the force field more focused in membranes. No? So I suppose that we have more history in this kind of system. I don't know. Is right?
Any further questions? If not, then let's thank Danilo again.
Thank you.